Okay, so welcome to this intensive revision session, which is our first revision session for the day. Okay, so in this particular session, we are going to focus on one of the most common and very essential human physiology essay questions or short notes questions. Okay, so I implore each and everybody of you to pay particular attention as we will be unveiling or uncovering certain key concepts with regards to the same questions that we'll look at this particular time in this very session. Okay, so make sure if you have a notebook, wherever you'll be writing from, if you have a question, please feel free to ask. Okay, so this session will be recorded and will be shared on our YouTube channel for further reference at all, if you might want it even in the near future for its relevance to your course. So, with regards to test one, physiology, cat one, topics include cellular physiology, include nerve physiology. You have synaptic and junctional transmission. You also have muscle physiology. You also have, I think, blood physiology. Though blood physiology is not usually part of test one coverage, but depending with how far the students and the lecturers have covered, it might be part of test one. So blood physiology is usually part of test two, but depending with the fluctuations on how you are learning or you may be learning, it can find itself in test one. Okay, so are we good to go now? All right, great. Okay, so we are going to start with some of the key questions, essay questions under cellular physiology. Okay, so this file has a lot of questions. So we'll just go to the specific questions which have to do with what you have covered. Okay, so we'll start from question number 11. So question number 11 um, is from cellular physiology, specifically transport across the cell membrane. If at all you watched our recorded lecture on transport across the cell membrane, then you are able to actually answer this particular question. So the question can be given to you that state the four types of passive. Okay, so the key here is passive transport. Okay, state the four types of passive transport mechanisms in humans. So I just want someone to give us the four types of passive transport mechanisms in humans. You can type or you can unmute and speak, or you can raise up your hand if you want to speak. The types of passive active transport. The types of passive active transport. The types of passive transport. Anyone? Any takers of that particular question before we can explain on it? Okay, someone has written diffusion, osmosis, facilitated diffusion. Okay, facilitated diffusion, osmosis, and okay, so those are good submissions. Okay, those are good submissions. So when we are talking about these passive active I mean passive transport mechanisms in humans, you must understand that these mechanisms actually get to allow substances, okay, they allow substances to move across what, across the cell membranes without requiring energy. That is the first key thing that you must understand, that the passive transport mechanisms do not involve energy. Okay, they do not involve energy, the passive transport mechanisms. So among the ones that we have, the types that we have what we call simple diffusion. Okay, we have what we call simple diffusion. Okay, so now when we're talking about simple diffusion, it just involves the movement of the particles from a region of an area which has a very higher concentration, okay, from higher concentration to a region of low concentration. Simple diffusion. Okay, so there are key things that you must not forget when you are describing or talking about simple diffusion. Okay, you must include the characteristics. I'm not saying that you need to write characteristics, but this is a guide of the important things that you should consider. Okay, so you must understand that simple diffusion does not involve energy expenditure. Okay, and then the particles actually in simple diffusion, they move down the concentration gradient. Okay, apart from that, simple diffusion does not involve any carrier proteins. Simple diffusion does not involve any carrier proteins. So examples of simple diffusion include oxygen, when oxygen is diffusing into the cells, as well as when carbon dioxide is diffusing out of the cells. That is simple diffusion. Then the second one is facilitated diffusion. Okay, we have facilitated diffusion. Now, this facilitated diffusion is also a passive kind of transport mechanism which involves the molecules, I mean the particles moving down their concentration gradient. But now the difference is that facilitated diffusion involves the carrier proteins or channels. Are we together? So facilitated diffusion involves the carrier proteins or channels. Okay. So now the key characteristics that distinguishes, or the key characteristics that make up facilitated diffusion are that the same way the same way simple diffusion never had the expenditure of energy, that is also what happens under facilitated diffusion. The same way molecules, I mean particles, were moving down the concentration gradient, that is how they are also moving in facilitated diffusion. Are we together? Then the only major difference between facilitated diffusion and simple diffusion is that in facilitated diffusion, you have the use of carrier proteins or channels. Are we together? Are we together up to this far? All right. Now, examples, okay, because you might be required to give some examples with regards to simple diffusion, facilitated diffusion. I've already given you examples of simple diffusion. Now, let's look at the examples of facilitated diffusion. We have glucose transport into the cells. Okay, glucose transport into the cells, uh, where we have the use of the GLUT transporters. You have GLUT1, you have GLUT2 in specific cells. Okay, so those transporters help transport glucose into the cell. Also, the transportation of amino acids into the cell. And also, we have ion channels. For example, the transportation of potassium down its concentration gradient. Remember, when we were talking about when we're talking about membrane potentials, that is also under cellular physiology. When we're talking about membrane potentials, we talked of what we call potassium leaky channels. Potassium leaky channels, where we say that those potassium leaky channels, they allow potassium to leave or to exit or to efflux out of the cell down the concentration gradient. Okay. Then the third example of with regards to the question that we are tackling is osmosis. Just like Rachel talked about it, we have osmosis. Now, osmosis is just the movement of water molecules. Okay, so osmosis, we have water molecules. So these water molecules are actually passing through a selectively permeable membrane. Okay, the reason why this is also considered as a passive transport mechanism is that it has those characteristics which are very peculiar to this kind of transport mechanisms. We have no energy expenditure. Um, the movement is down the concentration gradient, and osmosis helps in regulating fluid balance. Okay, but you must understand that one of the major key players in the regulation of fluid balance and cell volume is sodium. Okay, is sodium, very important. And the sodium potassium ATPase pump, just like we saw when we had the lecture on transport across the cell membrane. So examples include water absorption in the kidneys. You look at this when you do a certain topic. Then water movement into the cells during hypotonic conditions. Remember, we looked at hypotonic, hypertonic, okay, all those things. Then the last example is filtration. Okay, the last example is filtration. Now, what is this particular filtration? Filtration is just the movement of particles through the cell membrane, but now it occurs where? Under pressure. Okay, this occurs under pressure. Okay, so characteristics similar. The only difference is at the end. Okay, it also has no energy expenditure. The particles move down their concentration gradient. Then this one only has to do with the pressure difference. Okay, for example, blood filtration in the kidneys, that is during glomerular filtration. Okay, so that is it with that particular question. Are we good to go? Can we go to the next question, which is also the next question, which is under cellular physiology? Are we okay? Is everything is clear? You can comment if everything is clear so that we proceed. In fact, we are revising as it stands now. This is a total revision. Okay. So now, um, the next question, we'll talk about question, now we'll look at question 12. Okay, we'll talk about question 12. So question 12 says, with the aid of a well-labeled diagram, compare and contrast between temporal and spatial summation. You learned this, right? We covered this when we are doing membrane potentials. We are good. We learned this, right? We did learn about temporal and spatial summations. Okay. Now, remember when we talked about there is something that we talked about when we were looking at the membrane potentials. So when we were talking about the membrane potentials, we started by talking about resting membrane potential. Remember, then we went ahead and also learned how to calculate the nest potential. Okay, then that was when how now we moved from resting membrane potential. We started talking about graded potential. So under graded potential, we mentioned a few key things that if we are to bring the resting we said that if we are to bring the resting membrane potential to threshold, it means that we have to excite the cell. Okay, so in this case, that is what we call excitatory postsynaptic potential. Then when we want to move the resting membrane potential away from threshold, that involves the inhibitory postsynaptic potential. Okay, now we said that for us actually to grade the rest membrane potential until it reaches threshold, we need to have more of the excitatory postsynaptic potentials compared to the inhibitory postsynaptic potentials. Now, in order for us to achieve that, we have two major ways through which the graded potential can be achieved for us to reach threshold. That is where now we have temporal summation and spatial summation. Okay. So now in this case, you've been asked to, according to the question, you've been asked to use a well labeled diagram. So in when it comes to a labeled diagram, you only have to show actually how temporal summation occurs and just explain a bit, or and also how spatial summation occurs and explain a bit. Okay, so um, talking about temporal summation, when we talk about temporal summation, what happens is that temporal summation refers to the integration of multiple synaptic potentials over time, okay, resulting in what? In an action potential. Now, what happens is that you have one, okay, we assume so. This is a neuron, and then you also have a neuron like that, okay. So this neuron is the one that is going to be firing on this other neuron. Okay, don't mind my drawing. Okay, so what happens is that this one presynaptic, okay, this one is known as the presynaptic neuron. So this one presynaptic neuron will repeatedly stimulate one postsynaptic neuron, okay, in order for us to hit the threshold. Okay, now the key features of temporal summation is that number one, we have multiple synaptic inputs that have said will arrive at the same synapse here within a short period of time. Okay, then what will happen is that each of those inputs, synaptic inputs that will be arriving will actually generate small excitatory postsynaptic potentials. Okay, will be exciting bit by bit. Then these excitatory postsynaptic potentials will summate or will add up, and when they add up, they increase the what? The membrane potential. Okay, then once the membrane potential has been increased and the threshold has been reached, in neurons, it is negative 55 millivolts. So once that threshold has been reached, you'll find that uh, we, we are going to have an action potential being generated. Have we heard about temporal summation? Are we good to go? Can we now talk about spatial summation so that we can do as many questions as possible? All right. Now, when it comes to spatial summation, so spatial summation is also, it has to do with integration of multiple synaptic potentials. Now, this one is from different synapses on the same what? Same neuron. So in other words, in temporal, we had one presynaptic neuron. But now in spatial summation, what happens is that we have multiple, we have three, okay? We have three presynaptic neurons. So those three presynaptic neurons will fire simultaneously on one postsynaptic neuron. Okay, so you find that the key features of spatial summation is that multiple synaptic inputs arrive from different synapses, okay, on the same neuron. Then each of those will generate small what? Excitatory postsynaptic potentials. Then what will happen is that those EPSPs that are coming from different different synapses um, will actually in summate, and when they summate, they'll increase the what? The membrane potential. Are we together? Are we together? We'll actually we'll actually get to uh this was starting from here. Yeah. What do you want to do? Oh. In class, I was on it, so I had to switch off the mic, but it's recording. Are we together? All right. So now, if the threshold has been reached, the same will occur and action potential will be generated. Now, when it comes to the comparison of the two, is that both temporal and spatial summation, they will involve the integration of synaptic what? Potentials. The number two is that both two mechanisms contribute to the generation of an action potential. Okay, because we're trying to grade the potential. Okay, then lastly, both they require summation of EPSPs in order for us to reach threshold potential. Okay, but when it comes to the contrast or the difference is that when it comes to the time course, temporal summation occurs over a short period of time, while spatial occurs simultaneously. Okay, because under temporal we are only expecting from one presynaptic neuron, then from spatial we are expecting from different presynaptic neurons. Okay, okay, are we together now? We are done with that question. I'm sure we have been learned, we have been revised some uh, some principles that is under name physiology. Can we proceed to the next question? Can we proceed to the next question? Remember, all the questions that we are tackling right now, these short notes essay questions are under cellular physiology. Can we proceed to the next question, please? Yes, the question asked with the the aid of a diagram. Okay, the aid of a diagram. So when it comes to the aid of a diagram, there is a diagram which has to do with because that is found under graded potential. Okay, it's found under graded potential. So when we are grading the potential, this is the thing. When we are grading the potential, where is that question? Okay, the question is here. Okay, the question is here. So I tried first by illustrating how you can draw with regards to temporal summation. This is a drawing with regards to temporal summation. Then you also have for spatial summation, it involves the three presynaptic neurons. So there would have been another one here as well as another one here firing on the same postsynaptic neuron. Okay, because the goal of both of these is just to grade the potential or to increase the membrane potential so that threshold can be achieved. Okay, so the whole idea is that even as you draw these, you must be able to explain and you understand what is happening. By the way, it has seemed as though it's a very long question answering it, but it's very brief. But I did that in order for you people to have a clear and proper understanding of everything. Are we together? Christine, can we proceed? All right. Now, the other question is explain the physiological importance of what? Of the sodium potassium ATPase pump. Explain the physiological importance of the sodium potassium ATPase pump. Okay, so this is under cellular physiology. When you are doing transport across the cell membrane, you also talk about this because it is very, very peculiar in the topic of transport across the cell membrane. So now, the sodium potassium ATPase pump is also known as what? Um, uh, you can just write as sodium potassium pump. So this particular pump is very cardinal when it comes to cellular mechanisms, especially when it comes to the maintenance of ion balance and electrical potentials, including cell volume. It is very important um, pump. Okay. Now, what are the functions? Okay, what are the functions of the sodium potassium ATPase pump before we talk about the physiological relevance of this pump? Okay, number one is that remember when we talked about resting membrane potential, we we said that the resting membrane potential is usually maintained to be in the range of somewhere negative 70 millivolts to negative 90 millivolts. Okay, but the resting membrane potential of neurons is negative 70 millivolts. Now, the question was that how does the resting membrane potential remain or reach or stay in the range, in the normal range? Okay, so that is where now we talked about the three major key players in maintaining the resting membrane potential to be in the range. Okay, so we talked about the leaky sodium channels, we talked about the leaky potassium channels, and then we also talked about the sodium potassium pump ATPases. Okay, so that is where now the first function comes in. That number one, it helps in the maintenance of resting membrane potential. Okay, as it pumps three sodium ions out and two potassium ions into the cell. Then number two, it also helps in regulating ion balance. Okay, so what it does is that this pump actually maintains optimal intracellular and extracellular concentrations of sodium and potassium ions. By the way, I would also like to mention that potassium ions are the ones which regulate or play a key role in determining the resting membrane potential. Very important. Okay. Then it also helps in volume regulation. I think I mentioned this earlier. Okay, which has to do with cell volume. Okay. Now, when it comes to physiological importance, as far as our question is requiring us to answer in that direction, when it comes to physiological importance, is that number one, when it comes to nerve function, so it is important physiologically in nerve functioning, okay, because the sodium potassium ATPase pump enables nerve impulses by what? By maintaining the resting membrane potential and regulating the action potential generation. Then physiologically also it is important when it comes to muscle contraction, okay? Because this pump actually helps regulate muscle contraction and relaxation by controlling the availability of sodium and potassium ions. Remember that when it comes to muscle physiology, as we are going to see in a few questions as we proceed, is that sodium and potassium, they all take part in muscle contraction. Okay, because for the contraction of a skeletal muscle to occur, it first has to start from the beginning, and in that in the beginning or in the initial, you'll find that potassium and sodium, they play a role. Okay. Also, when it comes to cardiac function, the sodium potassium ATPase pump maintains what? Proper heart rhythm and contraction force. Okay. Also epithelial transport. Okay, so this pump also helps in ion transport across the what? Epithelial cells in the kidneys, in the gut, and the other tissues. So these are the response if you are to answer this particular question with regards to the physiological relevance or importance of the sodium potassium ATPase pump. That was that with regards to that question. Are we good to go to the next question? Can we proceed to the next question? All right. So we are going to skip all these questions and we are going to go to question number 21. I want us to exhaust cellular physiology essay questions before we go to other topics. So we are going to go to question number 21. Okay, so question number 21 says state the four types of active transport. So you see, in question number, was it 11? We talked about the active, the passive, passive transport mechanisms in humans. Now, this question is requiring us to talk about the what? The active transport mechanisms in this case. Okay. Now, when we're talking about active transport mechanisms, please, if you haven't watched our recorded lecture that we shared on YouTube on transport mechanisms, go and watch it. That is where we talked about everything that you need to know about the transport mechanisms across the cell membrane. Okay, so what happens is that the types of active transport mechanisms are actually classified by whether or not they use ATP directly. That is, they use energy directly, okay, or they use energy indirectly, okay. So with that regards, we have the main four active transport mechanisms are as follows, okay. We have primary active transport, okay. We have primary active transport. These are things that you people know and have learned. In fact, you are even revising as if you are learning. You are revising as if you are having a lecture. Okay. So This is the thing. When we are talking about active transport, you need to understand that some substances require energy for them to cross through the plasma membrane, often because they are moving from an area of what of lower concentration to an area of higher concentration. Okay, that is the thing. So now I want us to start by talking about primary active transport. When it comes to primary active transport, you'll find that this one moves ions across a cell membrane, and it creates a difference in charge across the what that membrane. So you'll find that primary active transport tends to um, it tends to use ATP directly. Okay, it tends to use ATP directly. That is primary active transport. It uses ATP directly. Okay, then we have secondary active transport. So this secondary active transport is active transport of small molecules. Okay, now what happens with secondary active transport is that it uses the energy of the electrochemical gradient which was created by the primary active transport system. Okay, so this one it uses energy directly. Then this one, which is secondary active transport, will use the energy that might have been generated by active primary active transport. Okay, then we have endocytosis. Okay, we have endocytosis. So endocytosis actually falls under vesicular, but we separate because they want four. The same way we have active transport, which is divided into primary and secondary. Okay, and then we have vesicular, where we have endocytosis and exocytosis. So endocytosis is a process by which a cell takes in substances from its environment, and it will form a vesicle around the material and then bring that vesicle inside the cell. Okay. Then we also have exocytosis. So now under exocytosis, that is where we have the movement of very large molecules across the cell membrane using vesicles from the inside of the cell to the extracellular environment. But now there is an alternative way through which we can talk about this same question. Okay, so the alternative way, for those who like a lot and big things. So the alternative way is that when we're talking about the primary active transport, is that of course it moves ions across a cell membrane and it creates a difference in charge across that membrane. You'll find that primary active transport system, they use ATP, okay, to move substances, which is true, such as an ion into the cell. Okay, but now you need to understand that the sodium potassium ATPase pump is a mechanism of active transport. This is an example that was covered in that lecture. Okay, just included it here for the sake of understanding. Okay, so what happens is that the sodium potassium ATPase pump is a mechanism of active transport. So it moves sodium ions out of the cell against the concentration gradient. Remember, sodium is more outside the cell than inside the cell. So for it to move outside the cell, it means it goes against what? Against its concentration gradient, right? So so it is with the potassium. Potassium is more inside than outside. So when potassium is being moved inside, it is moving against the concentration gradient. So there will be need for energy to push those ions against the concentration what gradients. Okay, so you'll find that both ions, the sodium and potassium, actually move from their areas of lower to higher concentration. So you'll find that the energy is needed for this uphill process.
and that energy is provided by ATP, which is adenosine triphosphate. Is that clear on primary active transport? Then we also have the secondary active transport. So under secondary active transport is that this one describes the movement of the material or of materials using the what, the energy of the electrochemical gradient. So this electrochemical gradient was established by primary active transport. Are we together? So you find that by using this energy, which was created by primary active transport, other substances such as amino acids, glucose, among others, can be brought into the cell through membrane channels. Okay. The other important thing that you should take note with regards to secondary active transport is that the ATP itself is formed through secondary active transport using hydrogen ion gradient in the mitochondrion. Okay. Then we have two types of vesicular transport, of which I have talked about. You have endocytosis. Okay, so endocytosis is a type of vesicular transport which moves a substance into the cell, and what happens is that the plasma membrane of the cell will actually engulf a substance, will engulf the substance, and a vesicle will pinch off from the membrane, and the vesicle will carry the substance into the cell. Now, what happens is that when an entire cell or other solid particle is engulfed, that is what we call phagocytosis. Okay? But now, when a fluid is the one that has been engulfed, that process is what we call pinocytosis. Okay? Then something that is peculiar of taking note as well is that when the content is taken in specifically with the help of receptors on the plasma membrane, that is what we call receptor mediated endocytosis. Then lastly, we have exocytosis. Okay, so exocytosis, here you'll find that a vesicle which contains the substance moves through the cytoplasm to the cell membrane. Then the vesicle membrane will fuse with the cell membrane, and the substance will be released outside, outside the cell. Are we together with that question? I think we have explained further on that, and people might not have any challenges with that. Are we good? Can we go to the next question? Can we proceed to the next question? All right. So we are going to jump to question number 24 of this file. So question number 4 is saying action potential is all or none in character and is said to obey the all or none law or principle. Explain how the action potential follows what we call all or none law. Okay. Now, you must understand that when we're talking about this all or none principle, the all or none law, okay, the all or none law is actually a principle which states that the strength of a response of a nerve cell or muscle fiber is not dependent upon the strength of the stimulus. Okay, so what happens is that if a stimulus is actually above a certain threshold value, a nerve or muscle fiber will fire. Okay, if it is below that, it won't fire. Okay, so according to this principle, according to this law, we'll either have a full response or will not have any response. Okay, now how does this O or none law work? So what happens is that if a stimulus is strong enough, an action potential will happen. Okay, and a neuron will be able to send information down the axon away from the cell body and towards the synapse. Okay, and you'll find that the changes in cell polarization will result in the signal being propagated down the length of the axon. Now, since you've been asked to describe or explain how the action potential is actually linked to the all or none law or principle, this is because an action potential will always be a full response. An action potential will always be a full response. What do we mean? This is because there won't be such a thing like no a strong or weak action potential. Okay, very important. You won't have a strong or weak action potential at any point. Instead, what will happen is that it is all or none process. It happens or it does not happen. Okay, so you can imagine if action potential uh, was like, okay, you can have a weak action potential. You'd find that if that weak action potential has been sent, it won't reach where it is supposed to reach. It might just end in the way. So, for the action potential being something that obeys the all or none law, it actually helps minimize the possibility of that information will be lost along the way. Are we together? Are we together? I need to know if we are together so that I proceed. Now, this process of the action potential is actually similar to the action of pressing the trigger of a gun. Okay, so you find that a very slight pressure on the trigger will not be sufficient and the gun will not fire. But you find that when you apply adequate pressure, the trigger will eventually fire the bullet. So what happens is that the speed and the force of the bullet are not affected by how hard you press or you pull the trigger. The gun will either fire or it will not fire. So you find that in this analogy of the gun and the bullet, the stimulus represents the force applied to trigger the gun, while the firing represents the action potential. Okay. Now, how the signal gets to triggers an action potential. In its normal resting state, you'll find that the inside of a neuron is around the 70 millivolts, okay, of which all of us know. But when activated by the stimulus, the membrane will begin to depolarize, causing ion channels to open. So as a result, you'll find that sodium ions will enter, okay, and then the action and the change will depolarize it. Polarization will occur along of the axon. Okay. Now, what happens is that once the cell depolarizes to the required threshold, the action potential will fire. Okay, because just as the all or none law states, this action is not graded. It is either it happens or it does not happen. Are we together? We've answered that particular question. Are we together? Can we proceed to the next question? Can we proceed to the next question? All right, we proceed. So we go to question number 25. Okay, so for question 25, I'll leave it up to you to look for it. So question 25 says, using a well-labeled diagram, state the ionic fluxes of sodium ion and potassium ion during the neuronal action potential. Okay. So question 25, I've left it for you. Let's check out question number 30. Still under cellular physiology. Hopefully things are making sense. Okay, so we are now looking at this question, question 30. Two more questions to finish cellular physiology revision, I'm sure. Um, so this question says, name the different body fluid compartments, the size of each, and the ways in which they can be measured. Okay, so when it comes to cellular fluids, you must understand that they are actually divided into two major functional compartments. Okay, two major functional compartments. Are we together? I believe we are all aware of those two major functional compartments. You have the intracellular fluid, which is the ICF, and then you also have the extracellular fluid, which is the ECF. So when we are talking about the intracellular fluid, when you're talking about the intracellular fluid, this is also known as the cytosol, okay, or cytoplasmic matrix. So the intracellular fluid is actually the liquid which is found inside the cell. So this particular compartment is just two-thirds, okay, two-thirds of total body water. So this two-thirds is actually so the two-thirds is 28 liters. Remember, total body water is 42 liters. So ICF takes 28 liters, okay, and the ICF is not measured directly. So we have the compartments which we can measure directly and those we can measure indirectly. Those that we can measure directly, we use dyes. Okay, we use a dye. Okay, but those that we can measure directly, there are calculations that you do based um, on the synchronicity of these particular compartments. So ICF cannot be measured directly, but it is measured by subtracting ECF from total body water. Then the second compartment is extracellular fluid. So the extracellular fluid actually, it is the fluid which is outside the cells. It is the fluid that is found outside the cells. Okay. Now, this extracellular fluid is the internal, okay? It is the internal environment of all multicellular animals. Now, ECF accounts only one third of total body water. So one third of total body water, which is 42 liters, ECF only gets 14 liters. Okay. Now, you must understand this same information that I'm giving you, you'll find this same information required of you when it comes to MCQs. Okay, so ECF is measured directly using radioactive sodium or sulfate or non-metabolizable, okay, non-metabolizable saccharides. So examples of these non-metabolizable saccharides, we have inulin, you have mannitol, you have raffinose, and you have sucrose. Okay. Now, what happens is that this compartment is further divided, okay, into other subcompartments. We have interstitial fluid. So interstitial fluid is neither ECF nor ICF. Okay, that is, the interstitial fluid is actually found, or it is the fluid that is found in intercellular spaces, and it accounts 3 over 4 of ECF, that is amounting to 10.5 liters. For you to find the interstitial fluid, you must subtract plasma volume from ECF volume. Then we also have plasma. Okay, so plasma is just a yellowish liquid component of blood. I'm sure you did it when you are doing blood physiology in your lectures. Um, that normally holds the blood cells. Okay, in whole blood suspension. So in other words, it is the liquid part of blood that carries cells and proteins throughout the body. Now, this plasma, it makes up 55% of the body's total blood volume. But when it comes to the ECF, it only accounts of 1 over 4 of ECF volume, which is just 3.0 liters. This one is measured directly using Evans blue. So to measure plasma volume, you need Evans blue or radioactive iodine. Then we also have transcellular fluid. So when it comes to transcellular fluid, is just a small amount of fluid which exists in specific cavities of the body. Okay. In your anatomy, you did cavities, so you know those cavities, such as the eye, the central nervous system, and the joints. So this only accounts 0.5 of ECF volume. We are done. Are we together up to this far before we proceed to the other question? Okay, so this particular question was talking about the different body fluid compartments and their size and how we can determine them. Muchimba, is that okay? Can we proceed? All right. So I think we're going to jump. Let me see what is there on question 37 with regards to cellular physiology. Okay. This one, I'll also leave it to you. Um, it says, describe in detail the physiological functions of endoplasmic reticulum and the ribosomes. This one, I'll also leave it to you. Let's see what is there on question 39. Question 39. Where is it? Question 39. So, question 39 talks about the physiology of the cell membrane. Even this one, I'll leave it up to you. Okay, it wants you to talk about the physiology of the cell membrane. But okay, let's talk about it. So, when we're talking about the physiology of the cell membrane, we know that the cell membrane is also known as what? Is known as the plasma membrane. Okay, is also known as the plasma membrane. Now, what happens is that this plasma membrane actually is a very critical structure which surrounds the cell and it separates it from internal environment from the it separates the internal environment from the external environment. Okay. Now, this one, the cell membrane actually controls the passage of substances in and out of the cell. When you're talking about the cell membrane actually is primarily composed of what we call phospholipid bilayer. Okay, so this phospholipid bilayer has two layers of phospholipid molecules, and those molecules are oriented with the hydrophobic tails. This was your biology in first year. Okay, hydrophobic tails facing inward, and the hydrophilic heads facing outward. Okay, it is that same arrangement which forms a barrier that brings us to say that the cell membrane is selectively permeable. Okay, now embedded in within the phospholipid bilayer are various what proteins. So those proteins, we have what we call integral proteins. So the integral proteins are the ones which tend to span the membrane and they are involved in the transport of molecules across the cell membrane. Okay, cell signaling as well as the cell adhesion. Then we have the peripheral proteins, which are attached to the surface of the membrane, and they play a role in cell signaling and maintenance of cell shape. Now, the cell membrane also contains cholesterol. Okay, the cell membrane contains cholesterol molecules. So those cholesterol molecules, what they do actually is that they help stabilize the membrane structure and maintain fluidity. So fluidity of the cell membrane is dependent on cholesterol. Okay. Now, additionally, you need to understand that carbohydrates are also um, carbohydrates are also often attached to proteins and lipids. So when they are attached to proteins, we have what we call glycoproteins. Then when they are attached to lipids, we have what we call glycolipids. And they also take part in cell recognition and cell-to-cell interactions. Okay. Now, the other thing that you should also understand or take note is that several processes that are involved in the physiology of the cell membrane include the passive transport. Okay, we also have active transport. Okay, so that is it with the physiology of the cell membrane. We are good to go. So we are done with cellular physiology essential questions. Are we good?